This is lecture NS-3. In this lecture, we're going to be talking about the central nervous system and comparing it to the peripheral nervous system, although peripheral will mostly come next lecture. In this one, we're going to go over the organization of the central nervous system, starting with how it forms, the idea of the neural tube, the difference between gray and white matter and how they're arranged, and the concept of the blood-brain barrier. We'll talk some about the spinal cord and how it's organized, and then we'll spend a lot of our time on the brain, kind of working our way up, going through the brain stem and then midbrain, thalamus, hypothalamus, and the cerebrum, talking about what each area does and a little bit about how it's organized. Let's start by talking about the difference between central and peripheral nervous system. So the central nervous system, or CNS, is the brain and spinal cord. That's where decisions are made. So this is the area where information is brought to the brain and spinal cord. Here's where we process most of it and make any sort of decisions about how best to respond, and then send those decisions out to the rest of the peripheral nervous system and to the body. The peripheral nervous system, or PNS, is nerves, which are mostly bundles of axons, ganglia, which are collections of nerve cell bodies outside the central nervous system, and many of our sensory receptors. These are about transducing sensory data, transmitting those signals to the central nervous system, and carrying signals from the central nervous system back out to the body. In some areas, the peripheral nervous system has some processing capability. There are, in the ganglia, there's some interconnection that allows a little bit of work to be done. But to some degree, the peripheral nervous system can be thought of as just being about gathering information and bringing it to the central nervous system, and then taking information from the central nervous system and taking it out to the body. Now, in terms of how this is organized, here we're looking at an embryo just days old. And what we find is that our embryo is composed of several layers of cells. So if we took kind of a cross-section of the embryo, what you'd find is that we've got an outer ectoderm and an inner endoderm, which is pretty much around the digestive tract. And in the middle, you've got some amount of mesoderm. And each of these layers ends up forming something different. Many different things. The nervous system is going to come mostly from the mesoderm. So let's take a close-up view of kind of this top part right here. And we'd be looking at some mesoderm, like so. That's ectoderm. And one particular area of that ectoderm is going to sort of get set off. This is an area, this is along what's going to be the dorsal ridge, basically the back of this organism. This is the neural plate. And some of the areas on the side of it are also distinctive. So I'll sort of shade those in here. And what happens at one point during this development is that this neural plate sort of pouches in, like so. So that the neural plate part is forming this section here, and the borders come in like so. And at some point, this just seals off above this, pinching off that neural plate into its own little tube. This is now the neural tube. Those border regions get sort of separated here. these are actually going to end up becoming the peripheral nervous system. Whereas the neural tube will be the central nervous system. And this tube now runs across much of the length of the organism. So if we imagined looking at our sort of growing embryo here, this neural tube is now running up and down the length with our split off peripheral nervous system stuff sort of along the sides. It's got this hollow space running up the middle. So these are the cells of the neural tube. They come from this ectoderm, which otherwise will become skin. So this will now become skin. It's kind of interesting that the nervous system and the skin come from the same layer. Now, on this long view, this tube is going to end up going through some interesting development. Specifically, if we say this is the rostral end of the organism, where the head is going to be, this end of the tube up here is going to start swelling up around certain areas they call vesicles, little swellings in the tube. So up there at that end, we're going to end up swelling up around several vesicles. And these vesicles are actually going to end up becoming different parts of the brain. So I'll show an image in just a moment here about what each of those does. So let's take a look at that. This image shows on the left the first three vesicles that develop at the rostral end of the neural tube. Don't worry too much about the names. I'm not going to ask you to remember them. But the frontmost one is known as the prosencephalon, the middle one is the mesencephalon, and the back is the rhombencephalon. Then over time, those start subdividing into more. So we end up, you can see there, with 5 vesicles. The back one, the rhombencephalon, becomes the metencephalon and the myelencephalon, which altogether forms what we often think of as the brainstem, the medulla, cerebellum, and pons. The mesencephalon stays as the mesencephalon. The prosencephalon splits up into the diencephalon, which will become the thalamus, hypothalamus, and epithalamus. We're not going to worry about the epithalamus. And you notice there what it says eye cup. That's actually what becomes the retina of the eye. So interestingly, technically the retina at the back of the eye is a part of the brain. And then the very frontmost swelling, the telencephalon, is going to become the cerebrum, which will include some subcortical structures and then the cerebral cortex. It's probably worth noting here that the rest of the tube down below what's shown there in the rhombencephalon is still there, and that's actually what's going to become the spinal cord. Here we're looking at that neural tube from the side and looking at how it develops. In this image on the leftmost one, we're looking at a 25-day-old embryo. The blue there is the prosencephalon. The reddish-brown is the mesencephalon, and the purple is the rhombencephalon. So you can see that as those develop, the purple ends up becoming the metencephalon and myelencephalon, those brainstem structures of medulla, pons, and cerebellum. The midbrain from the mesencephalon, kind of there tucked under, and then you can see how the prosencephalon, splitting up into the diencephalon and telencephalon, really ends up swelling up enormously. Now, it's also worth noting, as we saw in the previous picture, that one is one that splits actually out into 2 separate swellings, which will end up becoming the left and right hemisphere, which is an important organizational function of the brain. Okay, let's talk a little bit about the idea of gray and white matter. So as we mentioned earlier in these lectures, gray matter is where you find cell bodies and dendrites. In a way, it's where the computation happens. Whereas white matter is mostly bundles of myelinated axons. It's called white matter because it looks kind of whitish because of the myelin. Now, the 2 images you see on your screen here actually, oddly, look kind of similar, although they're not the same thing at all. In fact, they're from completely different areas. This is the spinal cord, as it might look in a cross-section, and this is the brain, and this other one is the brain. And this other one is the brain, specifically a coronal section. So what you're seeing here with the spinal cord is what you might get if you did a cross-section like that through the spine. Now, the gray and white matter are actually located differently in these two. In the spinal cord, this stuff out here on the outside is the white matter. It's bundles of axons either moving up toward the brain or down away from the brain. In these areas back here, this part, called the dorsal columns. These dorsal columns, in here the axons are moving up toward the brain. This is carrying mostly various kinds of touch sensation from the body. In all these other areas, these anterior areas down here, and these lateral areas out here, what we have are mixes of ascending and descending column tracts of white matter. So in those areas, some of it's going up, some of it's going down, and it's organized by what kind of information it's carrying. Now the inside here, this stuff shown in the middle, is gray matter. Not shown on here, there should be a little central canal, the fluid-filled canal, which is the remnants of that central fluid-filled area of the neural tube. But the rest of this gray matter arranged around there is organized into several areas. These up here are the dorsal or posterior horns. Those are the areas where you actually find mostly cell bodies of some, well, not some cell bodies. So some sensory cell bodies. Whereas down here, in this area where it says gray, these are the ventral or anterior horns. And here is where you find cell bodies of what we call lower motor neurons. These are the neurons whose axons are going to go out and synapse onto skeletal muscle, allowing us to move muscles around. In a few areas of the spinal cord, like this one is, you have also what's known as a lateral horn. This contains cell bodies of sympathetic postganglionic neurons. Sorry, preganglionic neurons. We'll learn more about those in the next lecture when we talk about the peripheral nervous system. So, that covers what's going on in the spinal cord. Now, in the brain section over here, which is what we call a coronal section of the brain, meaning kind of up and down in the same plane as the face, the gray matter is mostly here on the outside. So this is gray matter of what we call the cortex. But there are also some gray matters deeper down. So this is also gray. These are what are called subcortical nuclei. Specifically, these are basal nuclei. And this gray in the middle is of some structures more like in the center of the brain, things like the thalamus. Now, you can also see that there are some dark areas. For example, here and here. Those are fluid-filled areas called ventricles that are the remnants of that central part of the neural tube. They actually are continuous with this central canal of the spinal cord. So here in the brain, we've got the gray matter on the outside in the cortex and in some deeper subcortical structures. And then this other stuff in the middle is white matter. And the white matter here is kind of coming in several different kinds. There's white matter fibers that go from one area of the cortex to another, what we sort of call association fibers. So those are from one area of the cortex to the next, to nearby ones. We have what are called commissural fibers. So these are examples of association fibers. Commissural fibers go from one side of the brain to the other. Most of those are going through this particular structure, right here, which is the corpus callosum. So you saw how that final ventricle that became the cerebrum split itself into a left and right ventricle. That's what's giving rise to this whole left and right hemisphere thing. That has some interesting characteristics. We'll talk about those later in this lecture when we talk more about the cerebrum and the cerebral cortex. Then finally, we have projecting fibers. These projecting fibers, you can see them here somewhat in white, are going from cortical areas to and from the spinal cord. So we've got both things going up from the spinal cord up to the cortex and other areas, like here in the thalamus, and down from the cortex down into the and other areas down into the spinal cord. That's basically how the brain is connected to the rest of the nervous system. So that's kind of the idea of gray versus white matter here. Now, as we go on through here, we're going to talk very briefly here about the blood-brain barrier, which we've mentioned a little bit earlier. Not shown on these diagrams, but the idea of the blood-brain barrier is that unlike in most of the body, where a lot of materials move very easily in and out of between the extracellular fluid and the blood, in the central nervous system, a lot more control is exercised over what can get in and out. And that's done by a combination of changes in the capillary membrane and the action of glial cells, like astrocytes and ependymal cells, which add a layer of filtration and control to what sort of things can move in and out. So water, gases, and some ions can move easily between cerebrospinal fluid and blood. But most other things, except glucose, which is allowed to move very easily, but most other things are highly controlled. Some don't move at all, and some only move when they are specifically allowed to. All right, so our next topic that we're going to talk about here is getting into the specific organization of the brain. We've talked about as much as I wanted to about the spinal cord here. So now what we're going to do is kind of go through the brain from the base up and talk about what different areas are for and how they're organized.